So the first question is, does the LDL receptor mRNA increase or decrease with low cholesterol in the cell? And the answer to that is quickly that the LDLR mRNA increases as a result of low cholesterol in the cell. And the way that this happens is at the endoplasmic reticulum that's in a cell, there is the SRBP protein bound to the SCAP and the INSIG. When the SRBP is bound to these two other proteins, it is in its inactive form. The SRBP protein is involved in a negative feedback loop with intercellular cholesterol levels. So if a cell recognizes that there is high cholesterol or higher cholesterol in the blood compared to the amount of cholesterol that's in the cell, aka we have low cholesterol, the SRBP protein will actually have to become activated. And this is because you'll see later on that it binds the promoter region of the LDLR DNA. So what's going to happen here is, as a result of the cell recognizing that it has low cholesterol and that there is higher cholesterol in the blood, this inactive form of the SRBP protein, which is bound to the SCAP and the INSIG, will travel to the Golgi. And according to the paper, only the SRBP protein and the SCAP protein go to the Golgi. The INSIG protein does not. Now what happens here is two proteolytic cleavages. Now what this is doing is eventually what it leads to is the activation of the N-terminal activation, or I should say the release of the N-terminal activation domain of the SRBP protein. So this N-terminal of the SRBP protein will be cleaved off, activated, and from here it can go to the promoter region of the LDL receptor DNA. So I'll just remove all of this so we can put that bigger. So we have the LDL promoter. And specifically, the N-terminal activation domain competent of the SRB protein binds to what's called the SRE1 region of the promoter. So I'll just write N-terminal of S protein. So it's short, so that's the N-terminal of the activation domain. And then here we'll have the TATA box. And in this case, it will have RNA polymerase 2 and the two transcription factors needed for transcription to occur. So this is what the promoter region of the LDL receptor protein would look like in response to low cholesterol that's inside the cell when the cell notices that there's higher cholesterol in the blood. And we need this to be bound to the SRE1 region in order for any of this to occur, because this transactivation complement domain is the one that activates the promoter region and increases the amount of LDLR mRNA produced or transcribed. So that's the first question. And the second question is, if we had two groups of cells, so we had one group of cells, another group that had really low cholesterol, or I mean, it doesn't really matter at all anyways. What's really being looked at here is what happens when we add the MK206 protein. What happens to the amount of LDL receptor in each of these cells, or each of these groups of cells? So after reading the introduction, we know that when you treat cells with MK206, it inhibits AKT. And the AKT signaling pathway also has an effect on the amount of SRBP protein that's made, which we just discussed earlier. As a result of inhibiting the AKT, the kinase in this situation, we actually increase the amount of SRBP protein. Now, from the first question, we know when this protein is activated, it binds the promoter region of the LDL receptor gene. So we don't have to draw, I mean, if I drew the whole thing out again, it would look like this, and you'd have, you know, the SRE1 region, and this, the N terminal would be here, and then you'd have, you know, a TATA box, and you'd have the polymerase and the transcription factors. So this whole thing would be working, right? The LDL receptor DNA would be transcribed to mRNA in this case if we treated the cells with MK206. Now, why is that? Well, we just showed right here is that MK206 inhibits AKT, which in turn, inhibiting AKT activates this protein, and when it's in its active form, the N-terminal will go to the LDL receptor's promoter region and increase the amount of LDL receptor transcription. So if we were to just look at that for a second and then erase it, if one group of cells didn't get any MK206 and one group of cells did, the expected results would be in relation to this group of cells, there would be less LDL receptor protein, and then more LDL receptor protein. And this was shown in figure 1 when they treated cells with the MK2206 inhibitor. When they put the results on a Western blot, the band for the cells that were treated with MK2206 was darker and more visible for the LDL receptor protein. There was more of a dark band in this group of cells compared to that one. So as you can see, in this group of cells, there will be more LDL receptor protein.